Greeting of Union Yo from Texas A&M University. Today I will present cross-modality and self-supervised protein embedding for compound protein affinity and contact predictions, working with Professor Yang Shen. The motivation of this project starts from drug discovery that we're trying to design drug for not only the single gene or protein, but for a system that consists of a lot of genes and proteins. We need to look into more than millions of the proteins and mining the chemical space of up to 10 to 60 compounds. And if we look at over 80% of the FDA-approved drugs that are protein compound interaction, these spaces are actually facing a combinatorial explosion, not to mention that we have to make lots of expensive measurements for each protein compound pairs. That presents a critical need of the computational prediction of the compound protein interactions, also known as CPI predictions. Here we come up to our contribution focused on two keywords, cross-modality learning and self-supervised learning. These two words are not fancy work. First, cross-modality learning actually reflects the fact that protein exists in different modality. It not only 1D sequence, but also fold into 3D structure to functions. This is rational to introduce multimodality of the protein to incorporate the structure awareness. And these involve how do different modality models perform at different tasks, and how do we integrate different modality of the models. Second keyword is self-supervised learning. That recalling we have the scale label data. A natural way to address it is to exploit the unlabeled data and motivate from the language self-supervised learning. We developed the 1D self-supervised model, and parallelly we also developed the 2D self-supervised models for the graph modality model. And finally, we probably integrate cross-modality pre-training to further boost the performance. Here comes our method. Our base models is deep affinity plus from the deep relations paper, as we mentioned before. The only change is that we replace the hierarchical attention with joint attentions. The compounds are represented as the chemical graph encoded by GCN because we empirically find that compound graph work much more better than the SMILE sequence. The main focus is on the protein part. We do have the single modality models for the protein that for the 1D sequence model, we use the amino acid sequence as the input, and for the 2D graph model, we use the predicted intra-protein content map as the protein input. And separately, we use the hierarchical RNN as the sequence encoder and a graph attention network as the graph encoder. Notice that our graph modality model are still structure-free because we use the structure are predicted by protein, but such kind of the structure inductive bias are encoded from the predicted algorithm as shown in the Raptor X. Beyond the individual modality model, we also have the combined of the multimodality model named as cross-modality model. One way for the cross-modality model is the concatenation, which simply concat the different modality output as a joint vector and then fit into a multi-layer perceptron. Such kind of the concatenations are simple to preserve the information as much as possible. The second cross-modality model is named as cross-interaction that we additionally introduce the information flow between individual modality encoder, and this mimics the physical regularization between the modality. To improve the quality of the representation learning in accordance to the label starvation problem, we introduced the self-supervised learning. For the 1D sequence modality, we introduced masked language modeling, which tried to mask certain residue and predict the missing one. And similarly, we introduced the graph completion for the 2D modality model, also trying to mask and predict the missing residue, but based on the topological knowledge. And finally, we can integrate them together by joint pre-training. Acknowledgement in the last. Thank you for listening.